So picture this, right? You are standing in your living room, or I don't know, maybe out in your garden, holding a watering can.
Oh, yeah. The classic weekend gardening routine.
Exactly. And you are staring down at a plant you've been tending to for, like, months.
Right.
You check the soil. You occasionally miss the leaves. You've put it in that perfect sunlit corner of the room.
You've done everything right.
You think you've done everything right. But then today, you notice something is just wrong.
Uh-oh.
Yeah. A leaf near the bottom has turned yellow.
The dreaded yellow leaf.
It is. And the edges are kind of starting to curl inward. And that stem, which used to be completely upright and strong, is drooping.
Just looking totally sad.
Right. And if you are anything like me, your immediate thought is, oh no, my plant is getting sick. I need to intervene right now before this turns into a crisis.
Well, yeah, because it's the most natural reaction in the world, right?
Yeah.
We rely on our eyes.
We do.
We see a structural change or, a color shift, and we assume we are witnessing the onset of a problem.
Right. Like we caught it early. We see the yellow leaf and we think, okay, I've caught the opening act here. I can fix this.
But that's exactly the illusion.
And that illusion is completely shattered by what we're looking at today.
Oh, absolutely shattered.
Because, spoiler alert, that yellow leaf, that drooping stem, that is not the plant entering a crisis.
No, not at all.
That is the plant already deep, deep into a crisis. I mean, the alarm bells have been ringing for a long, long time.
We just didn't have the biological hearing aids to pick up the sound, basically.
Exactly. And today, we are taking a deep dive into what is honestly an absolute paradigm shift in how we interact with the natural world.
It really is. It changes everything.
We're looking at a fascinating new paper. It's published in Advanced Science recently by a team of researchers at the Institute of Science Tokyo.
Yeah, incredible work coming out of there.
And they've essentially built this incredibly tiny, transparent nanofilm electrode.
Just impossibly thin.
Right. And it allows us to actually read the invisible, silent electrical language of plants.
And I think it's important to state right up front that this isn't just a minor improvement on existing tools.
No, it's not like they just made a slightly better thermometer or something.
Exactly. This fundamentally changes the philosophy of scientific measurement itself.
That's a huge statement.
It is, but it's true. We are moving from an era of extracting information by force to an era of integrating with the biological world so gently that the organism doesn't even know we are listening.
That is just, it gives me chills, honestly. And the implications of that shift are staggering.
From saving your houseplants to, completely rewiring global agriculture.
Right. But to really understand how this microscopic technology works, we need to unpack why our own eyes are so terrible at diagnosing plant health in the first place.
We have to start with the visual lag.
The visual lag. Let's get into that. Because that really proves we just can't trust our eyes, right?
The visual lag is basically the crux of the whole problem. Human perception is incredibly biased toward macro visual cues.
Like big physical changes.
Right. If something isn't changing shape or color on a scale of centimeters or inches, it barely registers to us.
We just don't see it.
But plant biology does not cater to human eyesight.
So let's break down what is actually happening when that leaf turns yellow. Because it's not just passively dying, right?
Oh, no, far from it. Chlorosis, which is the technical term for that yellowing, is often a highly active, desperate process.
A desperate process. I love that phrasing.
It is. When a plant experiences prolonged stress, say a severe lack of water or maybe a nutrient deficiency,
It realizes it's in trouble.
yeah, it realizes it can no longer sustain its entire structure.
So what does it do?
It begins to actively dismantle the chloroplasts in its older leaves.
And chloroplasts, those are
They're the engines of photosynthesis, and they contain chlorophyll, which of course gives leaves their green color.
So the green is literally being packed up and shipped out.
Exactly. The plant breaks down those cellular structures to salvage vital nutrients, like nitrogen, for example.
Right.
And then it reallocates them to the newer leaves or the root system just to keep the core of the organism alive.
So it's essentially triage.
It is a triage mechanism, exactly.
Like an ER doctor deciding what to save.
Yes. By the time enough chlorophyll has been degraded for your human eye to register a yellow tint,
The damage is done.
millions of cells have already been dismantled. The plant has been in a state of emergency for days, perhaps weeks.
Weeks.
Oh, yeah.
That just makes me think of an engine, honestly.
An engine.
Yeah, it's like driving down the highway and you think your car is fine because, you don't see any smoke.
I see where you're going with this.
Right. But internally, you've been out of oil for 30 miles.
Your dashboard warning light was broken the whole time.
Exactly. The dashboard light is broken, so the engine block is warping. The pistons are actively fusing to the cylinders. The metal is literally shredding itself apart.
And you have no idea.
None.
Yeah.
By the time the violent black smoke actually billows out from under the hood and you finally pull over,
The problem didn't just start.
right, the smoke isn't the problem. The smoke is the autopsy.
That is a very grim analogy,
Yeah.
but incredibly accurate.
It gets the point across, right?
It really does. The smoke is the lagging indicator of a catastrophic internal failure.
So the yellow leaf is the smoke.
Yes. And in the same way, when you finally see that drooping stem you mentioned earlier, you aren't seeing the onset of dehydration.
What am I seeing then?
You are seeing the complete loss of turgor pressure.
Turgor pressure.
Yeah, the internal water pressure that keeps plant cell walls rigid.
Oh.
By the time that drops, the plant has exhausted every single localized coping mechanism it has.
Wow.
And it is now undergoing systemic structural collapse.
So we basically walk around looking at our gardens or farms thinking the field looks green, therefore the field is healthy.
But the reality is we are completely out of sync with plant time.
We are always late to the emergency.
Always.
So, okay, if the visual evidence arrives too late, and these visual cues are basically just a biological post-mortem, how do we catch the crisis in real time? Because the plant must know signaling its distress much earlier.
It does. It recognizes the stress the moment the oil runs low, so to speak.
Just in a language we haven't been able to hear.
Precisely. And to hear it, we have to move away from the visible spectrum entirely.
We have to look at the electrical life of the flora.
Yes. The silent electrical language.
Okay, let's slow down here, because this is where a lot of preconceived notions about biology, my own included, start to break down.
It can be a hard concept to grasp at first.
Because when we talk about an electrical life or a signal, my mind immediately defaults to animals.
Yeah.
I think of a brain, I think of neurons firing.
A central nervous system.
Exactly. A command center that, senses pain and sends a message to your hand to pull away from a hot stove.
Right.
But plants don't have brains.
No, they don't.
They don't have a central nervous system.
They are entirely decentralized organisms. They don't have a single organ responsible for processing information.
So, let's unpack this. If a plant has no brain, no nervous system, and no central command center,
Right.
how do ions just shifting around actually constitute a signal?
Whoa.
Because isn't a signal supposed to be intentional? Like, I am thirsty, send out an alert.
That is a very common human-centric bias.
Really?
Yeah, we equate communication with consciousness.
Well, sure, it feels like they go hand in hand.
But in biology, a signal doesn't require a conscious brain formulating a thought.
What does it require then?
A signal simply requires an event, propagation, and a subsequent reaction.
An event, propagation, reaction.
Exactly. The intention is irrelevant. The mechanics are what matter.
Okay, walk me through those mechanics then. What happens electrically when a plant gets stressed?
Okay, let's say a caterpillar takes a massive bite out of a leaf.
A very classic plant stressor.
Right. Let's zoom in on the exact cells where that bite happened.
Okay, we're zoomed in.
The physical trauma ruptures the cell walls.
Makes sense.
This physical break immediately triggers the release of certain molecules. And interestingly, one of them is glutamate.
Wait, glutamate?
Yes.
Isn't that a neurotransmitter? Like in human brains?
It is the exact same neurotransmitter that excites neurons in the human brain.
Wait, really? Plants use the exact same chemicals we do.
They do. We share a lot of deep evolutionary machinery with them.
That is wild.
It is. Now, in the plant, this chemical release, the glutamate, triggers specialized proteins in the membranes of the neighboring undamaged cells.
The cells next door to the bite.
Right. And these proteins act as gates.
Okay.
When they are triggered by the glutamate, they open up, allowing ions, specifically calcium, potassium, and chloride ions, to flood rapidly across the cell membrane.
And ions are just charged particles.
Exactly.
So if you have a sudden rush of charged particles crossing a barrier,
You are literally generating an electrical current.
you're creating electricity.
Precisely. You change the electrical potential of that cell, it depolarizes.
Okay, so that's the event.
Yeah.
But what about the propagation?
Well, plant cells are connected to one another by microscopic channels.
Like little tunnels.
Yes, they're called plasmodesmata. Think of them as tiny tunnels passing through the thick cell walls, directly connecting the cytoplasm of one cell to the next.
So the electrical change doesn't just sit there in that one cell.
No, it sweeps forward.
How fast?
Very fast. The voltage change in the first cell forces the ion gates in the neighboring cell to open,
Which changes its voltage.
which triggers the next cell and the next and the next.
It's like a stadium wave.
Stadium wave, yes.
Like the individual fans, or in this case, the ions aren't running all the way around the entire stadium.
Right, they aren't moving through the whole plant.
They were just standing up and sitting down in their specific seats.
Exactly.
But the energy, the wave itself, ripples across the entire arena at a massive speed.
That is a brilliant way to visualize it. It is an action potential, a localized wave of electrical depolarization.
And it travels far.
Oh, it can travel through the plant's vascular system, the phloem, to reach the furthest roots or the highest leaves in a matter of seconds or minutes.
Seconds. That's incredibly fast for a plant.
It is. And when that electrical wave reaches a distant leaf, it triggers a reaction.
Right. The third part of your signal definition. React.
Exactly. So the wave hits a distant leaf, and it might cause that leaf to start producing defensive chemicals.
Like things that taste bad.
Yes, chemicals that taste bitter to caterpillars long before the caterpillar actually climbs up there.
That is incredible. It is a highly sophisticated, data-rich telemetry system happening right under our noses.
It's a dynamic, constantly communicating network.
So every time there is a sudden drop in temperature, or every time the soil moisture dips below a certain threshold,
The roots experience that stress.
the ion gates open.
And a stadium wave of electrical warnings ripples upward through the stem.
The plant knows it is in trouble instantly.
Instantly. And the scientific community has actually known about these electrical waves for decades.
Wait, really? Decades.
Yes, the existence of plant action potentials was theorized a very long time ago.
Well, which brings up a very frustrating mystery then.
I know where you're going with this.
Right. If we have known that plants are basically humming with electrical warnings,
Yeah.
why did it take until this recent breakthrough from Tokyo to actually build a practical tool to listen to them?
It's a great question.
If the plant is actively broadcasting a status report, why weren't we picking up the signal all along?
Because for decades, our attempts to measure the signal were they were so heavy-handed,
Too rough.
so clumsy that we completely corrupted the data. We have to look at the history of plant electrophysiology.
Okay, let's look at it.
It's plagued by a biological variation of the observer effect.
Oh, the observer effect. Like in physics,
Exactly like in physics.
where just by trying to look at a particle, you bounce light off of it and you change its behavior.
Right, you alter the thing you're trying to study.
So how do we alter the plants?
Dramatically. Think about what a traditional electrical sensor looks like.
Like a medical sensor.
Yeah. Historically, researchers used silver chloride electrodes, or silver chloride.
Good.
These are rigid metallic sensors.
Metal on a leaf.
Right. And to get them to pick up an electrical signal from biological tissue, you can't just rest them loosely on top.
You need a solid connection.
You have to ensure strong electrical contact.
So how do they do that? Tape.
They would use heavy conductive gels or sticky adhesives.
Like the kind they use for an EKG on a human chest in a hospital.
Yes, exactly like that.
But a human chest is not a leaf.
Not at all. A leaf is a delicate breathing organ.
Right.
When scientists slathered conductive gels onto a leaf, they were suffocating the stomata.
The stomata, those are the pores, right?
Yes, the microscopic pores the plant uses to take in carbon dioxide and release oxygen.
So it sounds like we were basically putting a heavy, sticky, light-blocking cast on a leaf and then wondering why the leaf was acting stressed.
That's exactly what we were doing. We were essentially putting a plastic bag over the plant's mouth to try and measure its breathing.
That is so counterproductive.
And it gets worse.
How does it get worse?
These older sensors were opaque. They blocked light.
Oh, so they couldn't photosynthesize.
Right, they cast shadows directly onto the photosynthetic cells beneath them.
Starving them of energy.
Furthermore, they were heavy. A metal electrode might weigh only a few grams, but to a delicate stem.
That's a massive, unnatural load.
Exactly.
So if I'm understanding the biological mechanism you explained earlier,
Yeah.
if you block a plant's primary energy source, you suffocate its pores, and you add a structural weight that physically pulls on its cell walls.
What is the plant gonna do?
It's going to freak out.
Yes.
The physical weight alone is going to cause a massive trauma response.
Exactly.
Ion gates are going to fly open, and a massive stadium wave is going to ripple out from the exact spot where you glued the sensor.
Which means the electrical signal the scientists were looking at on their monitors.
It wasn't the plant's natural baseline.
No, it wasn't the plant reacting to an incoming drought or a subtle temperature shift.
It was the plant screaming about the sensor.
Precisely. It is like trying to determine a marathon runner's baseline resting heart rate by making them wear a 40-pound lead vest covered in sharp edges.
You aren't measuring their resting state.
No, you are measuring their physiological panic to your instrument. The data was completely poisoned by the method of collection.
It's almost a tragic comedy.
It really is.
The harder we tried to listen, the more trauma we inflicted, which just generated more biological noise.
It obscured the very signals we wanted to hear.
And this fundamental flaw really highlights a deep-seated arrogance in how we used to approach measurement, doesn't it?
Arrogance is the perfect word for it.
We expected the plant to accommodate our clumsy tools.
And that arrogance becomes even more apparent when you look closely at the microscopic surface of the leaves we were trying to measure.
Okay, let's zoom in on the leaf surface.
Because the historical approach wasn't just to glue heavy things to the plant.
It wasn't.
No.
Yeah.
It was often to actively alter the plant to make the glue stick better.
Wait, what do you mean alter the plant?
Well, leaves are not flat pieces of glass, right?
Right.
When you look at vital agricultural crops, tomatoes, soybeans, eggplants, they are highly textured.
Oh yeah, tomato leaves are super fuzzy.
Yes. They are covered in these microscopic structures called trichomes.
Trichome. Yeah.
Trichomes are fascinating. To the naked eye, they just look like a fine fuzz on the stem or the leaf.
That's a little fuzz.
But under a microscope, they look like a dense forest of spikes and bulbs.
Spikes and bulbs, that sounds intense.
It's a defensive landscape. There are non-glandular trichomes, which act like physical spears to deter microscopic insects.
Literally physical spears.
Yes.
Yeah.
And there are glandular trichomes, which are like tiny water balloons filled with toxic or sticky oils.
Built-in chemical warfare.
Exactly, designed to trap pests or reflect harsh UV light.
They are an integral part of the plant's armor then.
Crucial armor. They regulate water loss, they control temperature, they fight off predators.
So an old-school scientist with a flat, rigid metal electrode.
A forest of tiny spikes in oil balloons is a nightmare.
Because you can't get it flat.
Exactly. You can't get a flat sensor to sit flush against the actual epidermis of the leaf if there's a microscopic forest in the way.
So what did the early researchers do?
They demanded that the natural world be smoothed out.
What does that mean?
The scientific literature is full of methodologies where scientists would use gentle abrasion to physically scrape or shave the trichomes off the leaf surface before attaching the electrode.
You are kidding me. They shaved the leaves.
Yes, physical abrasion.
It feels like we were asking a turtle to step out of its shell just so we could get a better read on its heart rate.
That's exactly what it was like.
We were deliberately injuring the patient to attach the heart monitor. We were destroying the subject to measure it.
Yes. They treated a complex, functional biological structure as if it were just an incidental inconvenience.
Just a bit of static.
A bit of static that needed to be cleaned up so the math would work out for their sensors.
It reminds me of a terrible thought experiment.
Oh, let's hear it.
Imagine trying to study the aerodynamic efficiency of a bird's natural flight patterns.
Okay.
But your tracking device is a heavy rectangular box.
Right.
So you pluck the feathers off the bird's back, glue a literal brick to it, and then you watch it plummet to the ground and say, ah, fascinating, this bird is a terrible flyer.
It's the exact same logic. The assumption that the world must be simplified or stripped of its defenses just so our tools can make sense of it.
It immediately invalidates the data.
Completely.
Yeah.
Because when you scrape away a trichome, you are rupturing cells.
Right. You're causing trauma.
You are initiating a localized flood of calcium ions. The plant's internal alarms are sounding before you even turn your machine on.
So you're back to measuring the scream instead of the whisper.
Exactly. The team at the Institute of Science Tokyo recognized this historical trap.
They saw the brick on the bird.
They did. They realized that every previous attempt was fighting a losing battle against the physics of the leaf.
The leaf was never going to be flat.
Never, so they completely inverted the problem.
How so?
They realized they couldn't force the leaf to adapt to the sensor anymore.
The instrument had to adapt to the leaf.
Yes. It had to become so fundamentally unobtrusive that it could bypass the observer effect entirely.
So it couldn't just be a smaller metal box.
No, it couldn't just be smaller. It had to be practically weightless.
Weightless.
It had to establish an electrical connection without crushing a single trichome, without blocking a single photon of light, and without restricting the stomata.
Which honestly sounds like a physics impossibility. How do you build a machine with zero footprint?
It sounds impossible, but that is exactly what they did.
Okay, let's dig into the actual mechanics of this breakthrough. The 70 nanometer solution.
Let's do it.
How did they do this?
To achieve this gentle interface, the Tokyo team engineered an ultra-thin, transparent nanofilm.
Nanofilm.
And the materials they chose are what make this possible. It is a composite made of single-walled carbon nanotubes embedded in an elastomer matrix.
Okay, let's unpack those terms for everyone listening, because the chemistry here is brilliant. It really is.
Let's start with the elastom. An elastomer is essentially a polymer that has elastic properties.
Right. Think of it like a microscopic, highly breathable rubber band.
Why is that specific, stretchy material so crucial?
Because a living leaf is not static.
It moves.
Throughout the day, yes. As a plant takes up water or loses it to evaporation, the individual cells expand and contract.
Oh, of course.
The entire leaf subtly changes shape.
So if you put a rigid film on top of a leaf?
The moment the leaf swells, the film will either snap or shear away from the surface.
It'll just pop right off.
Exactly. But an elastomer stretches and breathes with the biological tissue. It moves as the plant moves.
Okay, so the elastomer is the flexible scaffold, but rubber isn't exactly known for conducting electricity.
No, it's an insulator.
Right. So how does this rubber band read the stadium wave of ions?
That is where the single-walled carbon nanotubes come in.
Okay, carbon nanotubes.
A carbon nanotube is an extraordinary structure.
What are they exactly? Imagine taking a single layer of graphite, just a 1-atom thick sheet of carbon.
Like pencil lead, but 1 atom thick.
Yes, graphene. Now imagine rolling that sheet up into a seamless cylinder.
Okay, a tiny tube.
These tubes are unimaginably strong, and more importantly, they are highly conductive.
So they conduct electricity perfectly.
Yeah.
So you basically embed these microscopic conductive wires inside the stretchy rubber scaffold.
Precisely. And they create what's called a percolation network.
What does that mean?
It means that even when the elastomer stretches, the carbon nanotubes inside it are so long and tangled that they maintain physical contact with one another.
Oh, I see. So they don't disconnect when it stretches.
Exactly, keeping the electrical circuit intact regardless of how much the leaf flexes.
That is incredibly elegant engineering.
It is brilliant.
But the materials are only half the story. The dimension of this film is what truly breaks my brain.
The thickness.
Yes, the thickness is roughly 70 nanometers.
70 nanometers.
Let's put that scale into perspective for everyone listening, because it's hard to grasp.
It's very hard.
A single strand of human DNA is about 2.5 nanometers wide.
Okay.
If you take a single strand of hair from your head right now and hold it up to the light, that hair is roughly 80,000 nanometers thick.
80,000.
This sensor is 70.
Wow.
You could stack 1,000 of these sensors on top of each other, and they still wouldn't be as thick as a single hair.
It is at the very frontier of materials science. It is so thin that it is effectively invisible, not just to the human eye, but to the plant.
Because it lacks significant mass.
Right. It conforms to the topography of the leaf perfectly.
But wait, how do you even handle something that delicate?
It's tricky.
If I tried to pick up a 70 nanometer film with tweezers, wouldn't it just disintegrate?
Oh, absolutely. You couldn't use tweezers.
So how do you actually apply this to a messy, real-world, muddy, wind-blown leaf on a farm?
That is a crucial logistical question. You don't apply it like a sticker. No, films of this scale are often transferred using a water bath technique or a dissolvable substrate.
What dissolvable substrate?
Yeah. You might synthesize the incredibly fragile film onto a water-soluble backing.
Like those dissolvable packing peanuts.
Kind of, yeah. And then you place the backing onto the leaf, apply a tiny amount of water.
And the backing just melts away.
The backing melts away, and the nanofilm is left behind.
And as the water evaporates, capillary action just pulls the film down onto the surface.
Exactly. The attractive forces between the molecules pull the ultra-thin film down so tightly that it wraps around everything.
So it just drapes over those spikes and balloons we talked about?
It doesn't crush the microscopic forest of trichomes. It drapes perfectly into the valleys between them.
Like a microscopic fog settling over a mountain range.
That is a beautiful way to put it. It rests in the valleys without flattening the peaks.
And because it's largely made of transparent polymers and microscopic carbon networks, the sunlight passes right through it.
The leaf can still photosynthesize.
The stomata can still breathe underneath it.
Nothing is blocked.
And it doesn't require any suffocating adhesives to stay on, because the molecular forces at that incredibly thin scale are enough to hold it in place.
Which means, for the first time in the history of plant electrophysiology,
the plant does not know it is being measured.
The observer effect has been minimized to almost zero.
The interface is completely modest.
Yes.
So we finally have the perfect listening device. We have this invisible, weightless, gentle interface sitting comfortably on the wild, hairy surface of a living leaf.
We do.
The Tokyo team boots up their monitors. What do they actually see?
They saw the truth.
The truth.
Without the noise of a trauma response drowning everything out, they were able to clearly observe the subtle electrical waveform changes that precede physical damage.
They caught the stadium wave before the structural collapse.
Yes, they monitored plants exposed to various stressors, drops in temperature, chemical exposure, water deprivation.
And what did the data show?
They proved definitively that the electrical signal changes dramatically before any visible symptoms emerge.
So long before the leaf turns yellow.
They captured the internal warning long before the chlorophyll began to break down, long before the cell walls lost pressure.
So we are talking about time travel in agriculture.
Essentially, yes.
We are shifting our entire agricultural timeline.
Moving from reactive autopsies to real-time predictive forecasting.
Instead of waiting weeks to see a drooping stem, we can know within minutes that the internal machinery of the plant is struggling.
Minutes, exactly.
Let's extrapolate this. What does this technology look like when we scale it up? Like from a sterile lab in Tokyo to a massive industrial farm? Because, what does this all mean for the global food supply? It sounds like we're giving farmers a crystal ball.
We really are. And the logistics of scaling this are incredibly promising.
Really? Because putting 70 nanometer films on a million plants sounds hard.
Well, we aren't suggesting that a farmer needs to apply nanofilm to every single soybean in a 10,000-acre field.
Okay, good. That would be impossible.
Completely impossible. Instead, you utilize the concept of sentinel plants.
Sentinel plants? Okay, I like the sound of that. How does a sentinel plant work?
You strategically distribute these nanofilm sensors on a select few plants across different quadrants of a massive field.
It's like a canary in a coal mine.
Precisely. These sensors can be linked to tiny, lightweight microtransmitters.
How are they powered?
Perhaps powered by ambient radio waves or tiny solar cells, and they continuously monitor the local electrical potential of their host plant.
And they just wait for the stadium wave to hit.
Exactly. Now imagine a drone flying over the field on a routine patrol.
Okay, I'm picturing it.
Visually, its cameras see a perfectly uniform sea of green. The human eye sees a healthy crop.
The whole field looks fine.
But as the drone passes over, say, Sector 4G, it picks up a Bluetooth signal from a sentinel plant.
And what is this signal saying?
The nanofilm is reporting a specific, sustained calcium ion wave that correlates precisely with stomatal closure.
Stomatal closure. So the plant is closing its pores.
Which is the first internal defense against severe water stress.
So the plant is essentially saying, I am shutting down my pores because the soil moisture just hit a critical low.
Yes, but it still looks perfectly fine on the outside.
Right. It hasn't drooped yet.
The drone relays this telemetry back to the automated irrigation system.
And the system responds.
The system opens the valves exclusively for Sector 4G, delivering water precisely where and when it is needed.
Days before that stress compounds into physical damage.
Exactly.
The conservation implications of that are massive.
Absolutely massive.
We waste so much fresh water in global agriculture through blanket irrigation.
We water whole fields because we don't know exactly which parts are thirsty until it's too late.
Right. If we can target our response, the water savings alone could be revolutionary.
Not just water, but chemical application as well.
Like pesticides.
Yes. Consider pathogen defense. Fungal infections can wipe out entire harvests.
Yeah, blight is terrifying.
By the time a farmer sees the visible mold or the necrotic spots on the leaves,
it's too late.
The spores have likely spread everywhere.
But if the sentinel plant detects the electrical signature of an immune response the moment the first fungal spore attacks,
then the farmer can deploy a highly targeted, localized antifungal treatment to just one specific acre.
rather than blanket spraying the entire farm with thousands of gallons of harsh chemicals as a preventative measure.
It is the ultimate realization of precision agriculture.
It is the difference between carpet bombing a problem and using a surgical laser.
A perfect analogy.
And we need this precision now more than ever. Look at the state of the global food supply.
It's precarious.
We are operating with incredibly thin margins. We have severe climate instability, heat domes appearing out of nowhere, shifting frost patterns.
Unpredictable weather is the new normal.
If a sudden thermal shock hits a region, we don't have the luxury of waiting 2 weeks to see if the crop survived.
No, we don't.
This early warning system could literally be the difference between saving a localized harvest and experiencing a regional food shortage. It gives us the ability to dynamically manage a living ecosystem rather than just passively watching it react to the weather.
It's proactive instead of reactive.
Exactly. But while the agricultural applications are the most immediate and tangible, the researchers at the Institute of Science Tokyo have surfaced a concept here that extends far beyond farming.
I'm really glad you brought this up. Because as much as I love the logistics of drone-assisted soybean farming,
It is cool.
it is. But the underlying philosophy of this deep dive is what has really stuck with me. We've touched on it a few times, but I want to dive deep into the philosophy of thinner contact.
It is a vital concept. This breakthrough forces us to confront a hidden assumption that has largely driven human engineering and scientific measurements since the Industrial Revolution.
What assumption is that?
We have historically assumed that if a signal is difficult to perceive, the solution is always louder extraction.
We are so conditioned to think progress means brute force.
We really are.
It's like, if you can't hear the radio broadcast, build a massive towering antenna and pump more wattage through it.
Right.
If you can't dig the tunnel fast enough, build a louder, heavier drill with a larger motor.
Exactly. If the natural world is messy and hard to read, bulldoze the mess and force it into a geometry we understand.
Shave off the tree cones.
Shave off the trichomes. That mentality assumes that progress strictly requires dominance over the environment.
We thought we had to overpower the subject to understand it.
But what this nanofilm proves is a profound inversion of that logic.
How so?
The realization here is that some forms of profound knowledge do not arrive through increasing our power. They arrive through drastically reducing our interference.
That is almost poetic.
It is. We didn't need a heavier machine to pry the secrets out of the leaf.
The leaf was already speaking.
The electrical signals were pulsing through the phloem every single day.
The natural world was screaming its status report. We were just shouting over it with our clumsy instruments.
We had to learn how to stay out of the way. The interface only became functional when it became physically modest enough to let the world remain itself.
So we stopped shaving the trichomes.
We stopped suffocating the stomata.
We stopped demanding that the biological subject conform to the limitations of our metal tools.
And instead, we built a tool that submitted to the complex microscopic reality of the plant.
It's a shift from an era of extraction to an era of coexistence. We can finally listen without interrupting.
It really makes you wonder, doesn't it?
Wonder what?
How much data we've historically misunderstood across all scientific disciplines simply because our measuring tools were too heavy to let the organism act naturally.
Oh man, that is a humbling thought.
It is, and it serves as a beautiful blueprint for future engineering.
Because the best technology of the future might not be the most massive or the most forceful.
No, it might be the technology that is light enough to disappear entirely, leaving the organism pristine while still capturing its truth.
Leaving the organism pristine. Well, we have covered an incredible amount of territory today.
We really have.
We started by staring at a visually deceiving yellow leaf on a houseplant, realizing that by the time our eyes register a biological problem, we are looking at the autopsy of a crisis that started weeks ago.
Right. We explored the invisible stadium wave of ions, the calcium, potassium, and chloride that ripple through a plant's vascular system.
Proving that organisms without brains are still acutely aware of their environment and perfectly capable of instantaneous communication.
Exactly. We looked back at the tragic comedy of older electrophysiology, where scientists accidentally tortured plants with heavy metal sensors and suffocating gels.
Ruining their own data by ignoring the observer effect.
And we saw how the brilliant team at the Institute of Science Tokyo bypassed all of that arrogance.
By weaving conductive carbon nanotubes into a flexible elastomer, creating a 70-nanometer film that acts like a microscopic fog, resting gently around the plant's natural defenses.
A modest technology that gives us predictive telemetry, allowing us to manage the health of global agriculture in real time.
Conserving water, minimizing chemicals, and adapting to a volatile climate.
And teaching us all that true progress doesn't always require a bigger hammer.
Sometimes it just requires a thinner, gentler touch. Thank you for joining us on this deep dive. I know I will never look at a drooping stem or a fuzzy leaf the same way again.
And, as we reflect on this shift from brute force to gentle observation, I'd like to leave you with one final thought to ponder.
Let's hear it.
If individual plants have been constantly broadcasting these intricate, localized electrical narratives this entire time, and we are only just now building tools quiet enough to hear them,
Yeah.
what happens when we start applying the 70-nanometer contact to the immense interconnected fungal networks beneath the soil?
Oh, wow.
What other silent languages are pulsing through the forest floor right now, simply waiting for us to stop demanding they speak on our terms?